As mentioned a moment ago, Chapter 5 is going to kind of encapsulate how the nervous system and the endocrine system work by using different types of chemical messengers. So this is going to be the foundation of really putting together how the nervous system and the endocrine system work to kind of control or communicate with different parts of our body. Our objectives for this 5th chapter is we're going to give 3 major functional classes of chemical messengers. So we're going to talk about paracrine messengers, neurotransmitters, and hormones, and identify a specific type of cell which would release each, and then generally the effect which they would have at their target cell. Additionally, instead of looking at just the function, we're also going to look at the structure, because the structure dictates the function. So we'll simplify them with the function, then we'll look a little bit more at the structures and different messengers. The categories we're going to look at include amines, catecholamines, peptides, steroids, and echinocylins. And then we're going to see, well, based on their chemical structure, we can probably have an idea where they are made, how they're released, transported in the interstitial fluid, and signal transduction, which just means what the target cell is going to do. So I don't know if we'll get that to that today, but then from there on, we'll contrast the signal transduction mechanisms, or what we'll just go on now, call lipophilic or hydrophilic messengers, and then get into some diversity of our receptors. Up till now, we just said receptors are receptors, but receptors can act as channels, like gated channels. If you've been in lab, we saw that terminology. Some receptors may be enzymes, and some may be associated with a G protein. They're called G protein linked. So we're going to look at some various receptor structure and function. Additionally, then we'll go through and compare and contrast, as mentioned, the nervous versus the endocrine system. We'll go into explicit details, which kind of give a survey of the differences. That is what we are going to study, but first we want to introduce this term, called intercellular communication. On your own, I want you to take about 30 seconds and think about that phrase and see what it means. Intercellular communication is a form of, working backwards, communication between cells. So cell talking, if you will. But not by way of verbal communication, probably by what? Chemical or even electrical or thermal. Like cells, I'm trying to think of cells talk. Those would be different types of stimuli. We can mechanically talk too. Very good. The primary ones between cells are, as you guys mentioned, the chemical and the electrical, and the major ways that cells talk to one another. Chemically and electrically are the more prominent ones between cells of your body, within cells. The other types of things listed, like mechanical and thermal, are types of stimuli, so they'd be external, right, type of stimulus. So between cells, they tend to rely upon electrical impulses or various types of chemicals. So, to tie it together, we've got to remember where we've been, where we're going. And so, if I want two cells to talk to one another, one cell needs to have a what? A receptor, and the receiving cell should have a what? Effector. Even an effector, and even before then, it's going to have some level of control center, establishing set point for that cell. So the complexity of the control mechanism may not be real broad. It may be localized to within a single cell receiving a chemical message. So our cells, so we can't forget the role of the receptor, the control center, and the effector. So real quick, for example, if I think about the optic nerve in my eye, it would be sensitive to light. The control center would be my brain that says it's too bright, and the effector would be the muscles in your pupils to, right, constrict. So we can think about it globally like that. It'd be more minimal, like insulin binding to cells at your muscles. And then the receptor binds, be an insulin receptor, binds insulin. And then the control center would be within the cell, the muscle cell, and the muscle cell would express more glucose transporters. The effect would be putting up more glucose transporters. If the cell has more glucose transporters, that cell could, what could it do? By more glucose transporters, the cell could transport more glucose, right, for example. So it could be localized within a cell, is what I'm trying to acknowledge. But we can't put that Unit 1 material behind us, is where I'm now leading you right through. As noted, some cells may talk by using electrical communication. And this is going to call, so we can talk about it directly, direct intercellular communication, by way of our gap junctions. Do you remember talking about those? I had a test on it on Tuesday. I mentioned that charged particles could diffuse from one cell to another. Just simply mobilizing a molecule from one cell to another is a form of communication. And especially if these are ions. Ions carry what? Charge. So if I carry charge, I can carry electricity, which is another form of communication. So it's certainly present, but not going to be the focus of this unit, really. We will see direct cellular communication in this third unit, when we talk about how our smooth muscle cells are connected. And so, for example, in your digestive system, how all the muscle cells in your digestive system work in kind of harmony, if you will, in concert. It's because they are connected by gap junctions. Same thing for your heart. You can probably appreciate that better, how the whole heart works in concert or unison. It's because the cells are connected directly by gap junctions. So there's this direct cell-to-cell communication. So it's important, but we're going to focus more on this indirect form of communication from cells to cells. Indirectly, right, the cells are not physically connected together. And because they are not physically connected together, cytoplasmic contents can't be easily transferred from one cell to the other. And so therefore, there has to be something, an intermediate, and that's some type of messenger. Generically, we'll just call it a chemical messenger. On a week ago, we used the word ligand. When we talked about receptor-mediated, right? That'd be a chemical messenger, I could say a ligand. And so there has to be some type of chemical messenger. We'll differentiate, we're going to say hormone, neurotransmitter, whatever the case may be. So this is just a generic look at it. The chemical messenger has to come from some cell, and it has to do what? Go to another cell because it's there to transmit signals to talk, right? That our mode of communication in this case is chemical. And so one cell is talking, one cell is listening, if you will. But we know that any cell that needs to listen to a particular messenger better have a what? A receptor. So the secreting cell will manufacture the chemical messenger. The target cell, I should incorporate this terminology, the target cell is the cell which expresses the receptor. This is figure 5.1b in your textbook.
So the secretary, that the secretory cell creates the ligand.
The secreting cell would secrete the ligand. And again, this is super generic. We're looking at two little round cells, right? So the secreting cell could be a nerve cell. The secreting cell could be an endocrine gland, right? The secreting cell could be in your digestive system, right?
So is ligand the same as the chemical messenger?
Ligand could be synonymous with the chemical messenger. Or you could be really specific and a little more specific and say neurotransmitter, or hormone, or paracrine messenger. Or you could be even more specific and say acetylcholine, right? Or just go on with the examples So, yes, chemical messenger is another just kind of generic term for ligand. A chemical messenger is a ligand that binds to a specific receptor. Here the receptor is bound in the membrane. I want to broaden your horizon though a little bit. Not all receptors are located to the membrane, and we'll differentiate that, if not today, on Tuesday when we come back on the weekends. But if I use the word target cell, the receptor-bearing cell, that's going to respond. It's going to receive and respond. Hopefully, if it's built right, it'll respond. So the question is, what type of stimuli or ligands may there be? And we listed these when we were talking about intercellular communication. The first two, chemicals and gases, would be more a form of our intercellular, right, between cell communications. If you want to bracket these two, the chemicals and the gases. The other four, the light, physical force, temperature, kind of the membrane potential, but at least light, physical force, and temperature are types of external stimuli, but your target cell would still need a receptor. But if we're looking at between cells, there's probably some chemical or maybe a gas, like nitric oxide or something like that, coming from one cell to target cells. Are all receptors built the same? No, they're not. So does all receptors function the same? No. And so if we look at what's going on when we're trying to introduce this signal transduction cell-to-cell talking, it's really not very simple. But we're going to do our best to try to simplify it as best we can, okay? But the reason how target cells respond to various messengers, is because there are thousands of different specific types of receptors. Not only that, the intermediate things referred to as transducers are variable, right? For example, these things called kinases. There are hundreds of those. Phosphatases, GTPases. So you can see like the magnification of potential response by a target cell. The receptors activates a different transducer, may activate second messenger systems. Okay, so you can begin to see the complexity of where we're going. And if you start quantitating this, you could look at thousands or more different types of various pathways that a target cell may undergo when a particular chemical messenger or ligand is actually present. So instead of us studying tens of thousands of pathways, we're going to do our best to kind of simplify it kind of universally. Like, well, generally this happens, or usually this happens, or this is kind of shown to be right? Proven or, I guess, demonstrated by particular activities. And so what helps us a little bit is of all the different protein receptors that may be present, they're grouped into about 20 families. Okay? So just like your family unit might have multiple people in it, protein receptor families would have multiple types of proteins. So their structure will be variable, but their general effect is pretty similar. In this image, did I give it to you guys? No. It's okay if you don't have it. I just wanted to show some receptors maybe, for example, like we saw with just little gated ion channels. Some may be really complex, some may be strictly intracellular, so they're diverse. So we'll pick out a few important types of receptors that we'll talk about for the rest of the semester. So we certainly won't talk about all the receptor families. The types of extracellular messengers, again, you could say chemical messengers, ligands, but this is the first time I've used this term, extracellular. What does that mean? Outside of the cell. And so that between cell communication is due to a chemical released outside of a cell. So you could also use that same type of terminology. The types of extracellular messengers used for intercellular communication include neurotransmitters, hormones, a word we haven't used yet called neurohormones, and then paracrine and autocrine signaling. The first three items we will use for the duration of the semester after introducing them in this chapter. Neurohormones has an asterisk because I don't think it's properly addressed or like fully addressed as its own type of classification here. So it's a conglomerate of a neurotransmitter and a hormone. Paracrine and autocrine signaling will also just introduce, but we really won't give any examples other than just characterizing it. So what I want to do is, on your guided notes, you have some cells. I want you to label which cells would be the source cell, the stimulus cell, the target cell, and the receptor. And we are looking at specifically the first class called neurotransmitters. Neurotransmitters. Label on your worksheet where the source cell is, where the stimulus, target, and receptor is localized. Alright, so in this simplified image, figure 5.2c in your textbook, where is the source cell? The top, what color is it so we can? The yellow thing. What is that yellow thing? A nerve cell. Okay, so this could be it's a super simplified nerve cell that you would find in your brain, in your spinal cord, your cranial nerves, or your spinal nerves. Okay, so you can call them a neuron or a nerve. In this case, neurotransmitters come from nerve cells. Nerve cells are cells that have membranes and organelles and all those features which we've talked about so far, but really only one thing is shown here, right? There's certainly be more complex than that. If you've been in the lab, what else is in here? What else besides, what did we cover in channels and pumps and all those things? Okay, all kinds of myelin sheath may be present as well. Okay, so it's very diverse, but oversimplified here. So this is our source cell. What is the stimulus?
Red.
The red thing. Is it colored red in real life? No. What do we call this? We would call it a
Chemical messenger.
It is a chemical messenger, and specifically it is a neurotransmitter. So in this case, our stimulus is this red thing, which is representing a neurotransmitter, which is a chemical. It is a stimulus because it's going to affect a target cell. So in this image, the target cell is the little orange, round looking thing. We know it's the target because why? It expresses the receptor. In this case, the receptor happens to be membrane bound. Another important feature that we need to address about our nervous system and neurotransmitters is the proximity between the source and the target is usually really, really close, microscopically adjacent to one another. And so this interstitial space is microscopic. Does anybody know the name of that space? The synapse is the whole thing, and the space between would then be called the synaptic cleft. But yeah, that would work. So why don't you go ahead and label that interstitial space between the two as Anyway, that space between you would refer to as the synapse. Again, we will clarify this more in the nervous system, chapter 7 and 8, but we're just introducing it right now. And so nerve cells can communicate with target cells by releasing neurotransmitters. Very good. A specialized type of chemical messenger which is released into a synapse. And the receptor, the target's usually right adjacent to it. So if you think about two cells in your brain, they communicate by neurotransmitters. Another type of extracellular messenger is called hormones. You've probably heard this term before at least. I want you to take a second and label the source, the stimulus, the target, and the receptor. So in this image, where is the source cell? Also on the very top, right? So this is our source. Where is the stimulus?
The green.
The green thing. Where is the target cell?
The bottom.
Bottom
Right
left. Left. Why is this cell over here in the bottom right not?
There's no receptor.
We don't see a receptor expressed, so even though it's right next to a target, itself is not a target. So it doesn't have the appropriate receptor. And the receptor in this case is also membrane bound. It could just as well be, depending on the chemical, intracellular in the target. You can call this a stimulus, or you can label it a what? In this case, you would call it a hormone. Why would we not call it a neurotransmitter?
It's among the blood vessels?
It's the first thing doesn't look like a nerve, and what else?
It's among the blood vessels?
It's among the there will be blood vessels around your nerves too, though. We just happen to see this, so let's clarify that a little more. The messenger is not only is it among it, the messenger goes into it, right, and it gets transported, right, to the target. So that's what's going to characterize a hormone, a chemical messenger transported in the blood to reach a target which may be really far away from wherever the original source was. If you recall, the neurotransmitters are released in a microscopic synapse, right? So they're right adjacent, kind of jokingly called neighbors. However, the target and the source of the hormone may be really far away. So think about the pituitary gland. You've heard of that. Releasing its hormones into the blood may target the ovaries or the testes, right? So quite a far way away from the original source. So okay so far, the major differences? There's a conglomerate between the two, not fully addressed by your textbook, and so it's going to kind of piece some of these images together. And we call them neurohormones. How can we classify a neurohormone?
Hormone in the brain?
What's the source? A nerve, but its messenger goes in the blood. So it's can you see how it's a combination? So a nerve cell, some nerve cells can release, what do you call them, neurohormones or hormones or neurotransmitters. They release their messengers into the blood to approach the target, which is still probably very far away. So it's kind of odd where it doesn't release just into a synapse. So our source in this case would be a neuron, a nerve cell. The stimulus, you want, go ahead and call it a what? Combine them together, let's call it neurohormone. I think your book will just call them hormones because they are in the circulatory system. But I want to make sure we throw that prefix on there so we know that's coming from a nerve cell and not an endocrine gland. And the target is probably very far away, but it's still going to have a specific receptor. And the target cell is going to still do something. An example of a neurohormone we used way back in the beginning of the class. Remember we talked about oxytocin? Classic neurohormone. We'll get an endocrine system, we'll call it a hormone, but it actually comes from the posterior pituitary. Goes into the circulatory system. Antidiuretic hormone or vasopressin. Have y'all heard of that one before? You will. About a month. You'll hear about it and know all about it. So vasopressin or antidiuretic hormone, neurohormone that goes into the blood. What else? What am I missing? Adrenaline, epinephrine, norepinephrine. They come from the adrenal medulla, and they get purged into the circulatory system, which you've probably experienced if you ever drive down 35 or 40, trying to get here in class, and you feel that surge, right? So that's a neurohormone in your circulatory system, and you're worried about getting an erect or something.
Okay, so the difference between just a neurotransmitter and a neurohormone is that the neurohormones, it's a nerve cell too, like that goes through the bloodstream, and then neurotransmitter is like nerve cell to nerve cell.
The question was, what's the difference between a neurohormone and a neurotransmitter? The major difference is that a neurohormone goes into the circulatory system to affect a distant target.
Okay.
A neurotransmitter may not only just go nerve to nerve, it may go nerve to muscle or something like that. And so the difference there is that a neurotransmitter does not, by our definition, does not go into the blood. It goes into a synapse. And so the effector or the target's right there, right
Okay.
adjacent. So it's the distance between the source and the target.
And then just like just a hormone, not a neurohormone, just a hormone is like.
The difference between a hormone and a neurohormone is a hormone is secreted by not a nerve cell,
Okay.
by some specialized epithelial glands, like the pituitary gland or ovary or right, so on and so forth.
Okay.
So it's not a nerve, and it releases its messenger into the circulatory system. Good question. Additional questions or clarifications? So we have neurotransmitters, we have hormones, and now we're kind of meshing some together. We're going to call it a neurohormone. Additional types of messengers include paracrine messaging. And paracrine, it's going to look pretty darn similar to what? What does it look similar to? Looks, it is still a chemical, we're still in extracellular messengers. So paracrines are, in addition to neurotransmitters, neurohormones, and hormones, these are also an extracellular messenger. So what else, Riley? Yeah, I think you said it, but it looks similar to what? Was, yes, the previous three things we've listed, which one does this look like? Looks a little bit like neurotransmitters, right? Hopefully, because the messenger, which is an indirect form of communication, is a form of intercellular chemical messaging. The messenger is released from one cell, and the target is right adjacent to it. Doesn't go into the what? Doesn't go into, well, it doesn't go into the, what's missing here? We don't see it going to the circulatory system. We wouldn't call it a synapse because a secreting cell is not a nerve. And so paracrine signaling is some cell releasing a messenger, and the target is right adjacent to it. So described here as a neighboring cell. You'll see this, for example, in the immune response. Like your white blood cells might leak with messengers, might call in other cells for effect. Or even in the digestive system, releasing products to talk to the cells adjacent to them. So we really won't address paracrine messaging beyond just differentiating between the nervous system, between the neurotransmitters. But we should still see a theme. A secretory cell releases a stimulus chemical messenger to affect a target cell, which expresses a specific receptor. And then once the receptor receives the stimulus, the cell is going to have its own little integration center and then bring about some type of change or effect. Another type of messaging is referred to as autocrine messaging. Autocrine prefix tells us what? Self. Okay, so it's self-messaging. So some cells can produce a messenger. So the source is the what? The target. It expresses the receptor, and it is the target. And this too, we won't elaborate on this semester.
What would be an example of?
Still same thing with like the immune system, for example. Things that can release and respond to its message as well. Can you see that feedback loop? Is this intrinsic? Technically, yes. The reason I'm kind of like, is because this same autocrine type of messenger may be released by a paracrine cell. So those two cell types could be releasing the same messenger and affecting each other. And so this same molecule could be coming from a neighboring cell, which by that means would be extrinsic. And so I'm not sure, but I can see how you're coming to that by all means, because it's within itself. But I think because it's releasing an extracellular messenger and you're going through all this receptor feedback.
Out and back in.
Yeah, I'm gonna guess it's extrinsic, but that's an interesting question about its regulation. I'm not sure on that, but I could see how both sides could be argued if it's intrinsic or extrinsic. My gut reaction, there's a messenger extracellularly, right? Although it did originate in that cell, I would still call it, right, extrinsic. But I don't know for sure if that's the right way to classify that. That's a great question. Additional questions?
Oh, your recorder thing.
So we addressed neurotransmitters, hormones, neurohormones, and then briefly summarize paracrine versus autocrine signaling. And then so got kind of a thought question. So what's the best way to get this? And so to understand how a messenger affects the target, we really need to go through it and look at the actual chemical messenger. So the best way to do it is just look at the messenger or the ligand. Again, we're being generic, the neurotransmitter, the hormone, the neurohormone, paracrine, or the autocrine, or autocrine messenger. What you want to look at is something that's fundamentally we've been kind of addressing all semester. You need to look at the solubility. Is it more lipid soluble? Is it more water soluble? Okay. So you got to address those questions. And you can also look at the overall just structure of the molecule, which would influence the solubility. Once you look at the solubility of the molecule, you really, just saying hydrophilic, lipophilic is going to give you so much background information. That took us probably a couple weeks to really kind of summarize cell functions in Unit 1. So we're going to tie in some concepts from Unit 1 by summarizing whether a molecule is more lipid or more water soluble. So if we look at our lipid or more water solubility, what is this, right? The question is, what does this tell us? If I've got a table, do I have it? No. All right. So if I look at the solubility of a molecule, a messenger, a neurotransmitter, hormone, whatever, what do you think I could learn? Well, it would be just, so if I'm looking at, I'm sorry.
The probable location of the receptor, whether or not it's so.
Okay, very good. So in addition to just saying it's hydrophilic or lipophilic, we then now know at the target cell where that receptor is even located, in the membrane or in the intracellular fluid. What else could we take away? Well, so we're looking at the messenger, not the target. Okay, so the messenger itself, if I say it's hydrophilic or lipophilic, I now know at the target where the receptor is probably located. As I go back to that messenger and I look at it again, the structure, what else can I take away?
How it's going to be transported there.
How it's going to physically get transported to the target. If it goes in the blood, there's a lot of what? Water. And so if something travels in the blood, it probably needs to be water soluble, hydrophilic. If it's not hydrophilic, then we need to find some type of accommodation for it to make it soluble at that moment in time. Furthermore, we know where the receptor is. We know how it's transported in the interstitial fluid or the blood. What else can we take away about a chemical messenger? So we have an idea. We know the solubility. We have a pretty good guess about what it's made of. What are our major molecules, macromolecules, used for messaging? Probably proteins, which tend to be what? They're made of amino acids, and so most of our amino acids tend to be, solubility-wise, insoluble lipids. So if we think about where water is everywhere, so proteins tend to be more water-soluble. I can work backwards. If I know a protein secreted as a messenger, I know what about it? If I know it's a protein, it's secreted out of the cell, what else do I know? We know that it's water soluble. We don't know that, it's probably water soluble. But if I have a secreted protein, well, in unit 1, what else do I know? It was made where? Yes. The rough ER, modified in the Golgi, secreted by vesicles, by which process? Exocytosis, right? So you can really back up. If I say it's a protein messenger, the secreting cell probably made it in a rough ER, modified it in its own Golgi, packaged it in vesicles, secreted it, and now it's water soluble. Okay, so we'll repeat those specifics. But what I'm pulling in, hopefully, is if you learn the solubility of a molecule, you learn more than it's water soluble or not. Okay, so we can look at the chemical class, like you said. Well, it's probably a protein or amino acid or a steroid hormone or a lipid, or pick your choice, right? Additionally, if we know if it's a lipid or protein base, we know whether or not it can or cannot be stored in a cell, in a vesicle. We also know if it's stored in the cell, we know how it can be secreted. Or if it's not stored in the cell, we know also how it would get out of the cell. Transport, and there's an asterisk by blood, not all messengers go into the blood. So you could say just transport in the interstitial fluid, okay, whether or not it needs help or not. Additionally, first identified, the solubility of a messenger. You have a pretty good idea where the receptor is located at the target cell. And then if you know where the receptor is, you have a general idea of what's going to happen, or what's referred to as a signal transduction, how long it will take, how long it lasts, the reaction lasts, and then relatively how stable that messenger molecule is. That's a lot of information when you just say something is hydrophilic or lipophilic. So the solubility gives you a whole litany of information. So what we're going to go through is differentiate our lipophilic versus a hydrophilic molecules and address each of these. These headers are there in table 5.2. So if you want to make a reference to table 5.2. So we can't forget, how many times have I said we can't forget unit 1? We can't forget the function of our organelles. We just reviewed that our rough ER manufactures what? Proteins that will be secreted or incorporated in the membrane, but we're not looking at the membrane-bound receptors, we're looking at the extracellular messengers. And then the smooth ER manufactures for us what? Lipids. So our lipid messengers, probably made in the source cells, smooth ER. Okay. So let's go through and just kind of review this correlation. Although I'm kind of bombarding you with a bit of information, looking at a molecule, whether it's more hydrophilic or lipophilic, is the more simplistic way to look at it, contrasted to the structure of the messengers. And so I like to look at it this way. If we look at our lipid-soluble messengers, and I should have this for you in your table, so I'll walk through this in order. our lipid-soluble messengers, chemically speaking, include your steroids, like estrogens, testosterones, and so on and so forth. Econyloids, and another hormone called thyroid, your thyroid hormone. That one has an asterisk by it, 'cause it's kind of unique. So if you want to write down thyroid hormone and put an asterisk by it. So if I use the word lipophilic, is that okay? Before you scribble everything, lipophilic. Philic means loving. Lipo means fat or lipid loving. So these type of messengers are lipid soluble. What part of the target cell is lipid soluble? Stop scribbling everything down. Think with me. What part of the target cell is lipid soluble?
Membrane.
The membrane. And so if we know if these things can diffuse across the membrane or not. Okay. So now, these chemically speaking are your lipid soluble messengers, or steroids, econyloids, and thyroid hormones. They can, at the target cell, right, we're going to see diffuse across not only the target cell, but across the membrane of the releasing cell. So can we store our lipid soluble messengers? Can I make up a bunch of estrogen and store it, or make up a bunch of testosterone and store it in the vesicle? Why can't I store these things in vesicles? They'll leak out because the vesicles are made up of
Membrane.
lipids, which are membranes, which are lipids. And so trying to keep something lipid soluble in a lipid soluble containing thing is futile. It's just going to diffuse on out. So we cannot store any lipid soluble messengers, like your steroid hormones. And so what happens as soon as they are manufactured, they just simply diffuse out of the cell, out of the source cell. Does that make sense? You make it, we can't keep it, so it just diffuses out of the cell. These tend to be hormones, and so they do have to enter in the blood. But we said the blood has a lot of what? Water. These are lipophilic, or we could say hydro what? Phobic. And so if you think about water-lipid interactions, water and lipid do what to each other? Repel. And so without any type of extra mechanism, you could almost have like big fat steroid globules in your blood, like globules of estrogen, globules of testosterone. Are fat globules in your blood good?
No.
No. And so we want to overcome that mechanism. And so what your lipophilic messengers tend to do is bind to proteins in the what? In the blood. In the blood. These are carrier proteins. These are not what? Well, they do transport, they carry, but they're not the receptor. They're there just to transport, to carry. And so in your blood, not only do you have water, not only do you have red blood cells and white blood cells, you have proteins. You get them from your diet and et cetera. And proteins, we said, are more, which type of solubility? Water soluble. So we have an ability to transport lipid soluble molecules, or lipophilic molecules, just bind them to some protein. So that's all the protein does. It doesn't change the structure of the lipophilic messenger. It doesn't change the function. It doesn't do anything to it other than just physically take it from the secreting cell over to the target cell. Does that concept make a little bit of sense? That transport protein has to let go of the messenger in order for it to have an effect at the target cell. And so there's a lot of mechanisms that we'll study at the third unit to illustrate how proteins let go of molecules. But basically in your capillaries, there'll be gradients pushing that messenger out of the blood into your tissue. Okay? Once out of the blood plasma into the interstitial fluid, it can approach its target cell.
Where does the protein grab the hormone? Is it right at the membrane, like as soon as it enters the body?
The question is, where does this interaction happen? It could happen immediately. It could happen a few seconds after the messenger gets into the blood, but it's definitely in the blood plasma. For most circumstances, never say never or always, but think about, so your blood is continuously flowing, so think about water in a river. Okay, if you go tube on the Illinois, have I done that? I have not, so I hear it sounds fun, but so imagine the river flowing, and then there's a bunch of inner tubes ready to go. You could be the lipid soluble messenger, you could hop into the river and catch a tube, a transport protein right away, or you might have to bounce around and then catch one. And so the molecules, and so there aren't carrier proteins waiting, they're just flowing through, and they'll just collide and interact at some point, and so
the hormone can get into the blood, it just can't travel.
it can get into the blood, it just doesn't travel very well.
Okay.
Some lipophilic messengers may get in the blood, just like if it were me getting in the river, I would probably never, I'd be the one who would never find a tube, right? You're like, well, and then you get off, right? And so that could happen too. So the preferred transport is for them to be in a carrier protein, but they're not so repulsive that they're going to cause congestive heart failure or something like that. And so the primary mechanism is for them to bind to a transporter, but they can a little bit, they can diffuse just in there. But the preferred mechanism is to get on a carrier.
So they bind to the carrier, the protein carrier in the plasma, blood
Yes.
plasma, and
Yes.
then they're taken to whichever receptor they need to go to.
Yeah, they just go, they're distributed in your body, right? So it may be secreted from your pituitary and make it in your blood, and it goes through your heart, and it goes wherever. And at some point, it's going to reach a capillary. It may go down to your big toe and not be a receptor, or may go to your liver and be cleared. But hopefully, there's enough secreted that some will get to your targets. But there's no direct port other than the circulatory system. Like you don't have a direct to pituitary ovary, right? So the goal is just to produce enough that we are able to maybe get to target non-target tissues, but enough to affect the target. Did I answer your question? Okay.
You did.
Good questions. So if we look at our lipophilic messengers, they are bound to carrier proteins, and there's some nuances about it. If you want in your textbook, I certainly want to address that. But once, okay, so we said, well, what happens? The carrier protein has to dissociate the lipophilic messenger. Okay, so you have to hop out of the tube in the Illinois River, right, and then get off the bank to go wherever you were going. And so the hormone would do the same thing. It'll diffuse across the capillary cells in the interstitial fluid and diffuse right into targets. And as mentioned, probably non-targets too, but they wouldn't have an effect. And so, because these lipophilic messengers can diffuse across the cell membranes, you won't find very many lipophilic messenger receptors in the membrane. And they are localized inside of the target cell cytosol or the target cell's nucleus. And so they're intracellular receptors. What we're going to find is if a receptor is within the cytosol, it's going to have an effect at the nucleus, which is, what is in our nucleus? DNA. So we're going to do something to the DNA. And so generally leads up to transcription, which is production of what? Messenger RNA, and I'm sure there's variations where you can make any of the other RNAs as well. But if we make messenger RNA, we can then use it later to make a what? Or pretty darn quickly, we can use it to make a protein. So very indirectly, lipid-soluble messengers like steroids cause the target cells to make proteins, whatever it is. Skeletal muscles will make contractile proteins. Some proteins make receptor, right? All the tens of thousands of different types of proteins, right? Could be altered in expression. And so therefore, the lipid-soluble messengers, since they have to not only bind to the receptor and then transcription has to happen, and then maybe translation has to happen. How long do you think this takes? Fast?
No.
No, it's gonna take a few minutes or longer to have an effect at the target cell. The effect being transcription and then maybe hopefully translation as well. And so therefore, right, it takes a little bit long, right, the overall process takes a while, and because it takes a while, these molecules, these lipophilic messengers, steroid hormones, generally last in the circulatory system for a while. Okay? So it's described here having a longer relative half-life. This will hopefully help you, right, put it together if you refer to figure 5.7b in your textbook. An endocrine cell or endocrine gland is shown here because the bulk share of our lipophilic messengers are steroid hormones. And so they are transported in the blood. And I think this may answer your question, Riley, like where do they interact with the transporter, and do they all have to bind to a transporter, okay? And so the first thing is our lipophilic messengers, what is this arrow representing? It's simple diffusion, right? They just simply diffuse out. Do I have this image for you guys on the next page? No, I'm sorry. Make a reference to look up figure 5.7b, right, to complete this table. This is just summarizing the table. The lipophilic messengers are lipid soluble, so they simply diffuse out. They tend to go into the circulatory system, where, because they're lipophilic, tend to bind to carrier proteins. The question is, well, how much? Greater than 99%. I don't know how accurate that number is. But the bulk of them are bound to a carrier, whose job is just to make it more soluble in the water. But as noted, not all lipophilic messengers have to be bound to a carrier to get transported and pushed down the circulatory system. We transport it right throughout the body, and then it's not stored. It diffuses from the source cell, diffuses into the bloodstream. It binds to a protein in the blood plasma. Okay, all of which we just summarized in table 5.2. Once in the circulatory system, the messenger, we said, diffuses into the target, where you can see the receptor, in this case, is where? In the cytosol. And in this case, the receptor and the messenger are transported into the, what's this? Nucleus. Nucleus, and then what happens is we can do some transcription to make RNA, and then in a perfect little world, that messenger RNA goes out into the cytosol and is translation to make protein. So if you want to kind of step back, what's the purpose? Lipophilic messengers cause targets to make proteins. Whatever that protein is depends on the target cell. Your muscle cells make different proteins or express different proteins than your nerve cells. Then, right, we just keep going on the different tissues. So we just said the signal transduction is going to lead to transcription and then hopefully translation, but reality says that's not always a guarantee that it happens. What questions do you have at this point about when I say lipophilic messenger, how that's a loaded word? You okay at this point, or questions at this point? We can deduce the same type of information looking at hydrophilic messengers. So let's do that. The exact same thing. We can look at our chemical classes. I'll talk to this table using the images. So this should still be table 5.2 in your textbook. But these water-soluble or hydrophilic messengers may be fully built peptides or amino acids or even little tiny amine groups is what you'll find with these. Okay, so let's go back and look at this. So if I have a amino acid based messenger or a protein, that's secreted out of a cell, a source cell, we know that this was probably manufactured where? At the source cell's what? The rough ER. We said secreted proteins are made at a cell's rough ER. Stored in, I skipped a step. Modified in the Golgi, stored in vesicles, and secreted by exocytosis. So let's go through and look at that. Your hydrophilic messengers. Here we have an endocrine cell that could have just as well shown a nerve cell secreting a messenger across a synapse. Okay, but we'll look at, say, peptide hormone. So the endocrine cell, the hydrophilic messenger, they can be stored. So I can my body can make insulin and store it in things called vesicles. My body cannot make a bunch of estrogen and store it because it's what? Lipid soluble, lipophilic. But I can make some of these protein messengers and keep them around. Wait to use, get them ready to go, like the neurotransmitters in your nerve cells. They're ready to go. You don't have to wait for them to be manufactured. So they're made at the rough ER, modified at the Golgi, stored in vesicles. The process which they are released is called what? Exocytosis. Right, exocytosis from the source cell. They're hydrophilic, so do we need any specialized transporter for them? No. They've already got their tube on them. You ever go to the little river analogy, right? They're already water soluble. And so once exocytosed, they diffuse. Their mechanism getting into the capillary, remember we used the word transcytosis the other day? They may actually be transcytosed into the bloodstream. What does that word transcytosed mean? Including both endo and exocytosis, because we're moving something across the cell. So this little pink line is actually a bunch of individual cells. So if you wanted to draw little endocytotic vessels, pushing it across and then dumping it into the circulatory system. But once in the circulatory system or once into a synapse, they are very water soluble. So we don't need any extra carriers or anything like that for these. They are already water soluble. They can diffuse, right? Get in the blood, and then once in there, you can use the word dissolved. They're dissolved in the blood plasma. They're already water soluble. They'll be distributed through the body, and then they've got to be either filtered or transcytosed once they approach the what? Where are they going?
The target cell.
To the target. The same messenger, right, is going to have to bind to a receptor. Which cells, A, B, or C, would our hydrophilic messengers bind to? A. Would it bind to A? Yes. Would it bind to B? Yes. What? Just any hydrophilic messenger could bind to receptor A, receptor B. Could it bind to receptor C? No. Because this what type of messenger is this representing? A lipophilic. Okay, so a hydrophilic messenger wouldn't be able to get into the cell. So our hydrophilic messenger receptors at the target are located where? On the membrane, where the binding site is extracellular facing. Okay? It's still incorporated. It'd be an integral membrane protein for the target, but the binding site for the messenger is extracellular facing. Going back to our discussion about the diversity of our receptors, man, they can be really simple or really complex. And so what happens after that is depends upon what? the type of the receptor, the structure. It's of a